So, Dr. Yoder, thanks so much for stopping by and making time to talk to me today. Do you want to talk about where you're based right now?
Sure. I'm on the Faculty of Biology at Cal State Northridge. Um, that's the California State University campus in one of the Cal State campuses in Greater Los Angeles. Northridge is in the San Fernando Valley. So think Burbank or Encino. These are all actually parts of the city of Los Angeles, but they're sort of the suburbs of LA.
Gotcha. Yeah, and so you work on a bunch of different things, like including kind of coevolution and local adaptation, and a lot of your work is from kind of a genomic perspective. Do you want to talk about the paper that recently came out, or do you want to talk about anything?
Sure. Yeah, I could talk about the MNAT metasynthesis.
Yeah, that'd be cool.
This is a pretty long gestating project. But yeah, so my research interests are the evolution of interactions between species, especially mutualisms. And I've used kind of a lot of different methods, but I have tried to build up a portfolio in sort of population genomics in these contexts. One of the systems I work on is Joshua trees. I like your background.
Yeah, that wasn't an accident. I'm trying to make these backgrounds thematic.
But so the new MNAT paper is, like I said, kind of a long gestating project, but it grew out of a paper discussion club at the University of Minnesota that I was a part of when I was a postdoc there. And this is looking at specifically local adaptation, and it's addressing a question that kind of goes to the foundation of my research interests. So it's all very well and good to sort of say, oh, species interactions are really cool. You have things like snakes hunting toxic newts, and you have yucca moths doing really elaborate behaviors to actively pollinate flowers, and there's lots of really, really cool natural history. But as evolutionary biologists, we're primarily interested in how biodiversity came to be, and so being excited about species interactions kind of begs the question of how much they've actually contributed to diversification, to the formation to the origin of new species.
Yeah.
And so on the one hand, we have a large body of literature studying what we understand as kind of the first step to speciation, which is local adaptation. In species interactions, there are lots of cases we know where local adaptation has arisen because interacting species are experiencing somewhat different contexts, and that changes the costs and benefits of the interaction, or there are just coevolutionary processes like frequency-dependent selection that are actually prone to causing separate populations to kind of go off on their own trajectories. But there's another even larger body of research on local adaptation in response to the non-living environment. So precipitation, temperature, all of these gradients of things that we think of as kind of the bedrock or the stage on which evolution happens. And there's sort of there's a fair bit of back and forth about which of those has actually had more impact, and a lot of, I think, back and forth about how you would even make that comparison. So among other things, like one of the ways we think coevolution between interacting species might be more prone to drive divergent natural selection and local adaptation is if those interactions are taking place in different ecological contexts. So they change the cost and benefits of the interaction. So that's kind of, is that the biotic interaction? Is that the abiotic environment having the effect?
Right.
so so the folks in this journal club, and it was really me and Ryan Briscoe Runquist who were kind of batting around ideas for how we might tackle this. And she and I were interested in coming up with a question that we could address with a meta-analysis. And so this is a method that comes up a lot in a bunch of different fields of the sciences, where you have multiple studies that address similar questions and use sort of similar experimental designs, but they maybe are measuring different things, or they're working with different populations. They're different experiments. They're just, the form is similar.
Yeah.
And so there are formal methods for extracting the estimated effects from statistical tests as simple as a t-test or an ANOVA, and combining them all together in a framework where it doesn't matter whether in this experiment we measured growth rate, and in this experiment we measured fruit production, and in this experiment we measured egg production.
Yeah.
You could come up with generic effects, right, and ask whether they change based on the things that are being tested in the experiment, or other contextual stuff that you can add on top of what was published. And so Ryan and I were thinking about these questions and thinking about what the options would be in the breadth of reading we've been doing, and we realized that one of the ways to kind of tackle this abiotic versus biotic question would be to look at experiments that explicitly tested them together. There's a much smaller, it turns out, body of research where people have set up experiments where they test both the effect of a species interaction, like the presence or absence of a mutualist or competitor, and the effect of some sort of abiotic variable. Yeah. And we had someone in the group who had some experience with what are called metasyntheses, which is this is a class of methods that isn't widely used in the life sciences, but it's basically a very formalized, systematic way to do a literature review. And I'm actually familiar with it because I've worked with a social scientist in a field where it's much more common. But basically, if you have a body of text, and you particularly like a collection of research papers, and you want to try to say something about what they say in general, or look for patterns, you can do a classic literature review, which is very standard in our field. And then you're sort of doing a lot of reading and taking notes and trying to keep track of things. A metasynthesis takes that to the next level and says, okay, you need to be deliberate about how you do this. And so what you do is you read all of the papers once and you take notes, and you think about what sort of recurrent themes you have seen, and you create this annotation. And then you go back and read everything again and re-annotate based on this list of themes. And you may do that multiple times to kind of refine the description of what you're reading and what emergent things are coming out, so that you're basically, the first time you read everything, you might come up with a pretty good list of themes, but having that instrument to go back for the second reading, you may spot things that you didn't before. So we decided that we would try to do the meta-analysis and metasynthesis side by side on the same body of research. Cool. Yeah, and that's really the origin of this paper. And so we went out and looked for as many published papers as we could find that were doing this very specific kind of local adaptation study. And we assembled a body of papers that seemed like they were appropriate. And then we split into two groups, and one bunch of us went through the meta-analysis portion. That was where I did most of my work. And the other bunch of the group worked through the qualitative metasynthesis read these papers more times than I would ever care to.
Wow, that's impressive.
Yeah, it's a fairly work-intensive process. Even compared to, I did a lot of squinting at figures and trying to turn them into usable numbers for the meta-analysis, but it's not the same thing.
Mm-hmm. Yeah.
And so let's see. So the body of papers that we came up with, ultimately we found 91 that seemed like they were reporting appropriate experiments.
Mm-hmm.
And we did a couple. So for the meta-analysis, we did a couple more passes for could we confirm that it was the right form of experiment? Could we get data from it? So you can either there are tools that let you turn figures into numbers, basically, if you don't have a supplementary data table.
Yeah.
Or more often, hopefully, authors had posted supporting data, although some of these went back to before that was standard practice.
Right.
In a couple of cases, we were able to write to authors and get raw data, but responses on that kind of inquiry are pretty tricky to get sometimes.
Yeah.
And anyway, so that at the end of that, we ended up with let me see here. I want to make sure I have the numbers right. Yeah, 31 articles yielded usable data, and that represents about 62 experiments, because we had lots of papers that were reporting what we could treat as separate experiments.
Cool.
Yeah. And from those, in a meta-analysis framework, we could formally evaluate a subset of them in terms of local adaptation. So did they find evidence that the population that was tested in its home environment did better than other populations brought into that same environment? And we could also just check to see whether the different environments or biotic interaction conditions tested had what the total magnitude of fitness effect was. So regardless of who's local and who's foreign, if you're comparing this population or individuals from a population in one environment versus the other environment, how big is the fitness difference in whatever direction?
Right. And
that's the framework that lets us compare biotic and abiotic effects, because we can say this population here is experiencing two different biotic environments that has some standardized fitness effect, and the same population, what's the fitness effect of the abiotic environment that was tested in the same project? And so that, in that framework, that yields actually thousands of these fitness contrasts, although they're not entirely statistically independent because they're coming from the same source.
Right.
But that's a pretty good data set to work with.
Yeah, that's really cool.
And it's still, so this is pretty small as a local adaptation meta-analysis goes. This is far from the first time somebody has tried to do this with local adaptation studies in general. There are some really foundational ones from back when I was in grad school. That you can even today you could, if you were just looking for like local adaptation in general, you could get probably hundreds of studies in a similar project.
Yeah. But it's really important that you were looking for studies that were kind of testing both, local adaptation driven by biotic interactions and abiotic interactions, because of the file drawer effect, right?
That's right. Yeah. So a concern with these is that experiments where you don't see a significant effect just don't get published, and they're not out there to be collected in the meta-analysis.
Yeah, which is just an unfortunate, by just sociology of science kind of property of publications, is that it's hard to publish null results, and so we don't know about the null results. Yeah,
and I think that's less of a like damnation of science publishing than we sometimes frame it as, because you know, null results are important, but you have to demonstrate that the experiment was worth doing in the first place. Like, it's sort of trivial to come up with an experiment that doesn't have a result.
Right, right.
That's the question is, like, was there good reason to think that there should be a result?
Yeah.
So anyway, with this particular sort of much narrower body of studies, we're taking it as read that the authors have selected environments and species interactions that they at least suspect have similar effect sizes.
Yeah.
Because they put them in the same experiment. And
can we get into the natural history of like one example a little bit? Because I feel like there's a paper where either Seagraves and or Althoff like looked at that for yuccas, right? Like weren't they looking for kind of like what's the importance of geography versus biology?
So that was
that's
done in a phylogenetic framework, which is a very different kind of analysis. But yeah, in yuccas, the conclusion there. Oh, sorry.
Go ahead.
I mean, the conclusion there is that geographical isolation seems to be driving reproductive isolation more than the interaction with
the host
pollinators
or something,
or
the pollinators, yeah.
And that's consistent. So like, there's a pretty clear origin of this highly specialized yucca moth mutualism at the base of genus Yucca, right? And yucca is not unusually diverse considering its sort of broader phylogenetic context.
Sure.
So we don't see an uptick in species formation associated with that.
So you were looking specifically for papers where they were done in really diverse groups too, right? Or is that?
Well, we were just trying to find any papers that had done them.
Gotcha.
Or done both of these two kinds of environments. And so some of the things that actually emerge, and this is a good reason, or this is a nice insight that we get from the meta synthesis, the systematic reading of the papers.
Yeah.
Is that people think about these two different classes of environments very differently. And they get designed into experiments in different ways. So the abiotic environments tended to be sort of natural gradients, like how far up this mountain slope are you, or where are you in this rain shadow? The biotic interactions tended to be more binary, so the interacting species was present or not. And that's because in some sense that's often the only way that it's straightforward to manipulate these things.
Yeah, it's hard to do like an abundance gradient or something like that. Yeah, it would be harder.
And it's not clear also, even if you could construct that, always how valid it is. So the really common one is removal of competitors from experimental plots when you're testing local adaptation in plants. And okay, you could put varying numbers of competing plants beside your focal plant, but you have to have some idea about what's the like critical range of neighbor density to make a difference. And it starts to get complicated really quickly.
Yeah, for sure.
But what we do find is that there are measurable differences in some specific contexts in how strong the relative fitness impact of the biotic versus abiotic environments that folks are testing, how strong those are. And one of the things that was or one of the things that's sort of a big picture question that we were able to answer with this is comparing those standardized fitness effects for abiotic versus biotic environments along latitudes, because we have this long-standing observation going back to Darwin and Wallace that there's more species diversity in the tropics.
Yeah.
And it's unclear what the cause-effect relationship is, but lots of folks have suggested that there's a differing intensity of species interactions in those in tropical versus temperate environments.
Mm-hmm.
And what we do see is that the strength of the abiotic environment increases, or the fitness effect of abiotic factors increased slightly as you go north, and biotic decreased slightly as you go north. Abiotic gets stronger toward the poles, and biotic gets weaker, and that's consistent with this hypothesis anyway.
Yeah, that's really interesting. I mean, yeah, it's really, yeah, I guess we haven't really gotten into in the class like why you might expect kind of different rules to apply in different geographies, but that's a really cool kind of advanced concept that some of the gradients, that some of the patterns that we might observe are kind of due to, you know, slightly changing rules of the game as you move around. That's really cool. I don't want to like eat up your whole afternoon, but do you have time at all to talk about the Decolonized DNA Day stuff that you participated in recently, or is that too much of a like?
I mean,
it's another whole thing.
Yeah, I think we might not have really have time to go into that without making this substantially longer.
Okay, yeah, that's cool. I'll just send some links to people. Yeah.
Yeah, that's all online.
Cool. Yeah. Well, I don't want to eat up the rest of your afternoon, but thanks so much for taking some time and talking to me about this stuff. It was really cool to hear about your research and everything. And
yeah, of course. Yeah. Happy to help.
Awesome. Yeah. Thanks a lot, Jeremy.
Yeah, and thanks for having me. Have a good socially distanced semester.
Thanks. You too.